We're going to take a journey today, one that starts in your own medicine cabinet and ends in a lab where scientists are quite literally rewriting our genetic code. This is the story of how medicine's toolbox has evolved. So let's just dive right in.
So think about this for a second. An aspirin, right? You can grab it off the shelf. But then you have these new cancer therapies that are basically personalized living drugs. I mean, they both treat us, but how they actually work, they're on completely different planets. And that huge difference, that's exactly what we're going to break down.
All right, first up, let's talk about the absolute foundation of modern medicine, small molecules. These are the classic pills, the tablets. The stuff that's been the backbone of what doctors do for more than 100 years. This is tool number one.
So what exactly is a small molecule? Well, they're these tiny chemical compounds, and they're so small they can just slip right through a cell's outer wall. Think of it like a master key. It's small enough to get into pretty much any room, or in this case, any cell, and it can open a whole bunch of different locks. Now that's great, but it also means it might unlock a door you didn't mean to. And, well, that's where side effects come from.
And you see these things all the time. I mean, aspirin for your headache, statins to get your cholesterol in check, even some really targeted cancer drugs like imatinib. These little guys are the real workhorses of the pharmacy.
But, there's always a trade-off. The pros, they're easy to make, and you can just take them as a pill. Super convenient. The downside, though, goes back to that whole master key problem. They aren't always picky, which can cause those off-target side effects. And for really complex diseases, they're often just not sophisticated enough. And that limitation, that's precisely why science had to go looking for the next tool for the toolbox.
And that brings us to the next chapter. This is where the story shifts from convenience to absolute, laser-focused precision. This is where we started using biology itself to fight disease. Welcome to the age of biologics.
So if a small molecule is that master key, a biologic is more like a custom-made key that's been designed for one and only one specific lock. These things are huge, complex proteins made inside living cells. And because they're so big, they can't usually get inside our cells. So they do all their work on the outside, targeting things with just incredible accuracy.
And you're probably way more familiar with these than you even realize. Insulin, for example, the hormone that's a lifesaver for people with diabetes, that's a biologic. A lot of the newest cancer treatments use something called monoclonal antibodies to hunt down cancer cells with incredible precision. And yep, those are biologics too.
And here we go again, another trade-off. The big advantage of biologics is that specificity. It means fewer side effects, and we can go after diseases we never could before. But that complexity creates new problems. They're expensive to make, they usually have to be refrigerated, and because they're so big, you can't take them as a pill. They have to be injected.
Okay, so let's just put them side by side to make it super clear. Small molecules, they're tiny. They work inside your cells. You take them as pills, but they're not super specific. Biologics, they're massive, they work outside your cells, you get them as a shot, and they are incredibly specific. Two totally different tools for two totally different jobs. But what if the job isn't just to manage a disease, but to actually cure it, right at the source?
And this is where we get to the most revolutionary part of this whole story. We're moving beyond drugs that just nudge biology one way or another, to therapies that actually get in there and rewrite it. We're talking about, quite literally, living medicines. Okay, first up in this new world is cell therapy. So here, the medicine isn't a chemical, it's not a protein. The medicine is the cell itself. We can actually take living cells, sometimes engineer them a bit, and then use them to fight a disease. And the most famous example of this is CAR T therapy for cancer. And how this works is it's mind-blowing. Let me walk you through it. First, doctors take out some of a patient's own immune cells, their T cells. Second, they take those cells to a lab and genetically engineer them, giving them a special new receptor that's designed to spot that person's specific cancer. Third, they grow millions and millions of these new cancer-hunting supercells. And finally, they put this living drug back into the patient, where it goes on a search and destroy mission. Okay, now let's go even deeper. Let's talk gene therapy. If a small molecule is a key, and cell therapy is like a trained security guard, then gene therapy is like getting the actual blueprints to the building and just changing the lock's mechanism itself. The whole idea is to fix genetic diseases right at the source by correcting the broken gene.
And there are a few different ways they can pull this off.
You can just add in a working copy of a gene to replace the one that's broken. You can silence a gene that's causing trouble, or, and this is the famous one, with tools like CRISPR, you can perform what is basically microscopic surgery on the DNA itself, cutting out the bad part and fixing it right there. It's truly editing the source code of life.
And this brings us to the newest kid on the block, the tool that pretty much everyone became familiar with during the COVID-19 pandemic, mRNA therapy. Now, this approach doesn't change your DNA. Instead, it just gives your cells a temporary instruction manual, a little piece of messenger RNA that tells them how to make a specific protein to fight off a disease.
And that difference is so important. Think of it this way. Gene therapy is like a permanent software update for your DNA. You're hoping for a one-and-done cure. mRNA therapy, on the other hand, is like sending a temporary memo. It tells the cell what to do, the cell does it, and then the memo gets thrown away. It's totally reversible and has a very different set of risks. So wow, we've gone from simple pills to custom proteins to living cells to literally editing our own genetic code. The speed of this innovation is just staggering. So you gotta wonder, what's next? What's on the horizon?
Well, the field is just exploding with new stuff. We've got technologies like ASOs and RNA, which give us even more ways to control the messages our genes send out. CRISPR is getting more and more precise every day. And then there's synthetic biology, which is all about creating smart therapies, cells that can literally sense a disease and respond all on their own.
But you know, the real breakthrough, the future, it isn't just one of these technologies by itself. The future is all about putting them together. I mean, just imagine using AI to design the perfect mRNA instructions, delivering them with an engineered cell, all to treat a disease based on your specific genetic code. That right there, that's the future of personalized medicine.
You know, there's a quote that just wraps this whole journey up perfectly. Small molecules manage diseases. Biologics target diseases. Cell and gene therapies rewrite the rules of biology. It's such a powerful way to think about it, right? We've gone from just managing symptoms to fundamentally changing the game.
So we end with this question. We have now developed tools that honestly feel like they're straight out of science fiction. We can edit our genes, we can program our cells, we can design drugs atom by atom. Now that we actually have the power to rewrite the rules, the biggest question isn't just what can we do next, but what should we do next?